Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the cell cycle. Now, in this particular video, I want to focus on how the G2 phase of the cell cycle that ensures that the DNA replication is complete. If you look at the DNA and the active replication forks, during the active replication, you are going to see the single-stranded DNA because these active replication forks, they are going to generate these stretches of the single-stranded DNA for the replication. Now, during the S phase of the cell cycle, the single-stranded DNA regions, they are bound by a specific protein, which are known as the replication protein A. When we talk about these replication protein A or these RPA, this is actually a heterotrimeric protein complex made up of three proteins: the RPA1, the bigger subunit, which is 70 kilodalton, the RPA2, and the RPA3. So during the S phase of the cell cycle, the single-stranded DNA, they are bound by this particular replication protein A until they are replicated by the DNA polymerase. And the reason for binding of these RPA to the single-stranded DNA is to prevent the formation of the secondary structure or prevent the unwanted recombination events. So whenever there is a single-stranded DNA during the process of the DNA replication, these replication protein A, they are bound to these single-stranded DNA which are generated by the active replication forks. Now, when you move to the G2 phase of the cell cycle, and there are still single-stranded DNA or there are still active replication forks. So these single-stranded DNA, these RPA complexes, if they are there in the G2, that would mean there is some sort of problem going on with the replication, or there is some problem with the structure of the DNA, or there is damage to the DNA, or there is incomplete repair. So when there is ongoing replication, because we're talking about the cell division, so I'll be focusing on this ongoing replication. So if the G2 phase senses the presence of the single-stranded DNA RPA complexes, it is going to interrupt the cell division. And how it do is that it is going for the replication of a particular kinase, which is known as the ATR kinase. And this ATR kinase is actually activated by these RPA coated single-stranded DNA. So when there are single-stranded DNA attached to the RPA, that is indicating some problem that leads to the activation of this ATR kinase. Now, once you have activated the ATR kinase, what it is going to do? This ATR is going to prevent the entry into the mitosis. It is going to prevent the entry into the M phase of the cell cycle and hence the division of the cell. How it do this? So when the ATR it gets activated, which senses the ongoing replication or fork problems, what it do, the ATR, it goes for the activation of another kinase, which is known as the checkpoint kinase 1. How it do this? The ATR is going to phosphorylate this checkpoint kinase 1 at specific serine residues, which is the serine 317 and serine 345. So simply you can say that the ATR is a kinase. It phosphorylates the checkpoint kinase 1, thereby activating it. Now, when the checkpoint kinase 1 it gets activated, what it do, because this is a kinase, that would mean it is going to act on its substrate by adding the phosphate group into it. So the substrate for the checkpoint kinase 1 is a phosphatase, which is known as the CDC25. And when the checkpoint kinase 1 phosphorylates the CDC25 by phosphorylating it, it is going to make the CDC25 inactive. It inhibits the activity of the CDC25. Now, the CDC25, this is very important if you want to move into the M phase of the cell cycle. And once it is inhibited, that would mean you are not able to move into the M phase of the cell cycle. Now, what the CDC25 do is it is responsible for the activation of the CDK1 Cyclin B complexes, which is actually the mitotic trigger. But if the checkpoint kinase 1 phosphorylates the CDC25, you are going to prevent the activation of this mitotic trigger. You are stuck in the G2 phase of the cell cycle. What this means is that you need to do something about this. So when there is the ATR, CHK1 kinase signaling, that would mean you are arrested in the G2 phase of the cell cycle. You cannot move. Once you have replicated the DNA, once you have completely replicated DNA, you have got rid of the replication forks, you should move into the M phase of the cell cycle. But the first thing you need to do is you have to take care of this ATR checkpoint kinase 1. How it is happening? When the replication that is complete, you have to go for the decline of the ATR and the CHK1 signaling pathway. Now, once the replication forks that are resolved, all of the replication that has been completed, that would mean that there are no single-stranded DNA now. When there are no single-stranded DNA, that would mean that the RPA would not be able to bind to the DNA. When the RPA is not able to bind to the DNA because there is no single-stranded DNA now, that would mean that the RPA would not be able to go for the activation of the ATR kinase. When the ATR kinase that is not activated, that would mean that the CHK1 that is going to become inactivated, because it is the ATR that activates the CHK1 protein. When the CHK1 that is not activated, that would mean there will be a drop in the ATR CHK1 activity, and that would indicate that all of the replication forks, they are now completed. There are no single stranded DNA region present now, and the DNA replication that has been completed. Now, once the DNA replication that has been completed, that is, you can say, a sign that you can now move into the M phase of the cell cycle, and you can go for the division of the nucleus as well as the cytoplasm. So when the DNA replication is complete, and if you want to move from the G2 into the M phase of the cell cycle, you need to go for the activation of the CDC25. Now, the CDC25, as I've told you, this was inhibited by the CHK1. Now, because the CHK1 that has been inactivated, that would mean that the CDC25 will now be active. The active CDC25 is going to go for the activation of the CDK1 cyclin B complex. And when the CDK1 cyclin B complex that is activated, that would mean you can now progress into the M phase of the cell cycle. Now, in my next video, I'll be talking about how the CDC25 that is responsible for the activation of the CDK1 cyclin B complexes, and hence progression to the mitosis.